notice it was the same. Pretty bad. All right, so this is 22. This is going to be your fatty acid triacylglycerol metabolism. After this chapter, we're actually going to skip a bunch. Okay, and we'll skip forward to 28, I believe. And then we'll try to cover as much of that 28, 29, and on as we can. So this is the last one. You gotta have fatty acid and triacyl metabolism. Okay. Especially with you guys heard of like ketogenic and diets and stuff like that? Yeah. This is what it acts on, actually. You'll notice there's a lot of times where it's going to talk about fasting. That's actually what you're trying to mimic when you're doing those diets, in case you're wondering. Right. So the fatty acid physiological roles, fatty acids are going to have 4 major physiological roles. Fatty acids are fuel molecules. Fatty acids are going to be building blocks of phospholipids and glycolipids, i.e. cells. Fatty acids are going to have many proteins which are going to be modified by the covalent attachment of your fatty acids. These are going to function to target proteins to membrane locations. Without them, they wouldn't bind to the membrane, right? Fatty acid derivatives are going to serve as hormones and intracellular messengers. By the way, if you have a fatty acid imbalance and your body isn't making certain fatty acids, say for certain hormones, anybody have any idea what sort of effects you could see? If you go a shit ton of them, then you're right. I mean, you could have mental illnesses, you could have death, outright death. You could have an explosive reduction in your ability to heal. You could, an unbelievable number of things could happen. And very quickly, right? Because it doesn't take much hormone to have a large effect, right? So the triacylglycerols, these are going to be neutral fats, triglycerides. These are going to be the uncharged esters of fatty acids with glycerol. They're stored mainly in the adipose tissue. They're going to be stored also by the muscle, typically for energy needs. And this is why, for example, if you have breakdown of muscle over time, you start getting fat on your biceps and legs and stuff, right? That's that storage. Okay. And that's a triacylglycerol, by the way. And you can see how that's the neutral ester there, right? Okay, so the adipose tissue, this is going to be fuel-rich white tissue. This is going to be located throughout the body, notably under the skin. This is typically referred to as subcutaneous fat, and surrounding internal organs. This would be known as visceral fat, typically. The adipocytes, these are going to be fat cells that make up the adipose tissue. Major site of the triglycerol accumulation. The specialized for the triacyl synthesis, storage, and mobilization into fuel for the rest of the body. So this is actually an electron micrograph of an adipocyte. And we can actually see enormous intracellular lipid droplets, which are pretty kind of awesome. Ooh, so good. Okay, so the lipid droplets, these are going to be large globules that are going to be formed by coalescence of the triacylglycerols. These may occupy most of the adipocyte volume. These are going to be surrounded by phospholipid monolayer and proteins that are required for the lipid metabolism. Are they going to be water soluble? And that's why they have a monolayer, right? The phospholipid monolayer is what makes them quote-unquote water-soluble, though they won't actually be dissolved in it. It'll be like in a suspension, more like milk, okay? Typically. Actually, milk is very close. It's actually pretty much the same thing. So the triacylglycerols is going to be highly concentrated stores of metabolic energy. These are going to be reduced in anhydrous. Complete oxidation of fatty acids is about 38 kilojoules per gram compared to 17 kilojoules per gram for something like a carbohydrate or a protein. That's why if you look at the back of your containers, the fatty acids, fats, have larger energy numbers. A gram of nearly anhydrous fat actually stores 6.75 times as much energy as a gram of hydrated glycogen. And so this is why you store the vast majority of your energy in that form, right? Most of your cells can use that energy fairly easily, and it is far more energy dense. Imagine storing an equivalent amount of fat as glycogen and how much weight that would occupy and how much space that would occupy. Huge beer belly, right? Okay. So dietary lipids are going to be digested by the pancreatic lipases. Lipases are going to be intestinal enzymes that are going to degrade the triacylglycerols to free fatty acids and monoacylglycerol. Secreted by the pancreas. This right here is your triacylglycerol. Notice the three green things. Those are your acyl groups. You may recognize those acyl groups. Okay. Yes, we love the acyl groups, don't we? All right, so this is going to be your lipase. This is going to remove each acyl group one by one, and it's going to do that through hydrolyzing. Now, notice that's actually the same sort of reaction that you may recognize from back in organic chemistry. Remember the hydrolyze esters? No, that's that. So this would be triacylglycerol, diacylglycerol, monoacylglycerol, and that's where it usually ends. Okay? So the lipids are going to exit the stomach as an emulsion. The particles with the triacylglycerol core that are then surrounded by cholesterol and cholesterol esters. Bile acids are going to be the amphipathic molecules that will facilitate the lipid digestion by lipases. These are actually synthesized from cholesterol in the liver and secreted from the gallbladder. They orient ester bonds to make them more accessible to digestion by the lipases in the aqueous solution. And so you can kind of think of the bile acids a bit like your soap. Okay? It's going to break up those oil grease stains, if you will, and allow you better access to them. Okay? It's not actually that dissimilar from detergents, which is kind of interesting. Yeah, it's why, for example, if you ever want your detergents to work better, you put a little vinegar in there, a little acid, right? Make it acidic. Anyways, colipase, this is a protein that's going to bind lipase to the particle to permit lipid degradation. It's secreted by the pancreas. This right here is a glycolate. And you notice that this is a cholesterol-derived, what's that part? Carboxylate. If this was not in a neutral buffered environment, this could have been a carboxylic acid, right? Yeah. So that's a cholesterol-derived acid there. So in the intestine, the triacylglycerols is going to be reformed from free fatty acids and monoacylglycerol. It's going to be packaged into a lipoprotein particles. These are called chylomicrons. Now, one of the things I'd like to point out at this point is it seems very strange, and sometimes people have some difficulties with the fact that, yes, you're going to disassemble these, pass these through into your internal system, and then reassemble. Okay? The problem is that they're not small and they're not aqueous, right? They're not water soluble by themselves usually. And so you just cannot easily put these into the blood without any processing, okay? It's going to take effort. So the chylomicrons is going to eventually enter the blood so the triacylglycerols can be absorbed by the tissues. The free fatty acids and monoacylglycerols is going to be your lipase digestion products. Is going to be transported in micelles to the plasma membrane of the intestinal epithelial cells. The fatty acid transport proteins, typically referred to as FATPs, these are going to be membrane protein transports that are going to transport free fatty acids and monoacylglycerols inside your membrane. Remember, again, these are not trivially sized, right? They're fairly large, okay? The fatty acid binding proteins, these are going to be FABPs. These are going to be proteins that are going to ferry the free fatty acids and monoacylglycerols to the cytosolic face of the smooth ER. These are going to be then resynthesized, and then we're going to make with the resynthesis the triacylglycerols. Okay? So we got break it apart, transport it across, reassemble. Okay? Anybody ever know how they make a ship? Like the big destroyers? Like aircraft carriers, destroyers, all that stuff? No? Or any like the big windmill stuff, that sort of thing? Okay. So typically how they do those sort of things, or like the assembly houses that you can put up within a single day, they make the parts, they assemble them, they break them back down, and then they transport it one wall, one section at a time. And then they reassemble it back where it's going to be. Okay? And they do that because you would not want to assemble that in the dry dock and send it off into the water, and you don't know if the parts are fit together, right? Or put together a house and you need it to be functional in the next day. And whoops, this one was outside of tolerance. Now one of the walls doesn't come together. And so you basically end up doing the same thing here. To transport it more simply, you need to break it apart. The chylomicron formation and degradation, your chylomicrons is going to be lipoprotein transport particles. These are going to be composed of newly synthesized triacylglycerols with proteins, phospholipids, and cholesterol on the surface. They're released into the blood. The degradation of the membrane-bound lipases at the adipose tissues and muscles. And you kind of actually know chylomicrons more intuitively than you'd think. Okay? We've actually worked in a number of cases now in things like organic chemistry, but in several other situations, we've been talking about this in various different classes for chemistry, where you have these suspensions, right? Okay, that's going to be what a chylomicron ultimately is, okay? It's going to be a suspension where you make something a bit more soluble on the surface so that you can suspend it for long enough to do stuff with, right? Okay. And if you ever notice, if you ever take like a person's, have you guys ever seen plasma or blood that's been sitting around too long and it's went quote unquote bad? No? Okay. If you ever watch that, it actually separates in layers. And that's because, have you ever seen milk that's set around in your fridge too long? And like it gets a layer on the bottom that's that really concentrated white, and then you get the clear. It's like that. Anyways, so this is the fat absorption and transport. So here you have the lumen. This is going to have your triacylglycerols. You're going to hydrolyze them using the lipases. And again, we have a process to go through that make that more efficient, right? This is going to give you your fatty acids and a monoacylglycerol. Typically 2 fatty acids and a monoacylglycerol. This is then going to use the FATP. This is going to then transport that across the intestinal cell. The FABP is going to bind to those, make them easier to move around, i.e. ferry them, if you will. Though I often think using words like ferry is kind of weird in biochemistry because there's no intention to it, right? It's a random diffusion process. So your ferry is going like this. You'd be pissed. Okay, so this is going to then take those and reassemble them into your triacylglycerols. Remember, you got those components split apart, you transport them, you reassemble them right here in this area. The phospholipids, cholesterols, and proteins are going to join those. These are going to make your chylomicrons. Again, these are your TAGs in the center here. Notice you're going to have a more polar layer on the outside to interact with the water, a less polar layer on the inside. This is technically a mycel, okay? This is a mycel, actually. It's just a very small one, right? This is then going to exit. This is going to go out to the lymph system. From the lymph system, it will get exchanged into blood to feed all of your other stuff, okay? Stage 1 is going to be mobilization. So you're going to have mobilization of your lipids through triacetyl degradation and release from the adipose tissues and transport to the energy-requiring tissues. Stage 2 is going to be the activation and transport. So your activation of your fatty acids and transport into the mitochondria, where you can then harvest energy. Guess what stage 3 is? Harvesting energy. And so you're going to break that down. What was the best way to get energy out of these things? By the way, this is actually where remember how I had the 3 pathways and they were coming together? And they came together essentially in the acetyl-CoA or the pyruvate section. Eventually, they went down to the acetyl-CoA. This is that right there. So this is the fatty acids going into the acetyl-CoA, where they can then enter the citric acid cycle, right? And so now we're down there. Again, that's going to be degradation of the fatty acids into the acetyl-CoAs. Then we're going to process those in the citric acid cycle / electron transport chain and obtain the energy from those. Okay. The hormonally controlled lipases are going to catalyze the hydrolysis of the triacylglycerols to fatty acids. The glucagon and epinephrine, these are going to act through the 7-TM receptors. These are going to lead to the phosphorylation of 2 proteins of pKa. We have the paralipin, which is a fat droplet that's associated protein. We have the hormone-sensitive lipase as well. And so again, these are the triacylglycerols being hydrolyzed by the lipase. Though note that in this case, we go fully to glycerol and 3 fatty acids. It's not the monoacyl form, okay? Okay, so some of the effects of the phosphorylated paralipin. The phosphorylated paralipin is going to restructure the fat droplet so the triacylglycerols are more accessible to mobilization, i.e. breaking up. The phosphorylation of paralipin is going to trigger the release of a coactivator for the adipose triglyceride lipase, which is ATGL. The ATGL, which is again the adipose triglyceride lipase, is going to initiate the mobilization by releasing a fatty acid from your triacylglycerol, forming a diglycerol, and then bound to its coactivator. The hormone-sensitive lipase is going to convert the diacylglycerol into a free fatty acid and the monoacylglycerol. This is that normal monoacylglycerol we've been talking about before. Okay, so we're down to that point. The monoacylglycerol lipase is then going to convert the monoacylglycerol into a free fatty acid and a glycerol. This will complete your fatty acid mobilization, and now instead of having the monoacylglycerol, we have another free fatty acid, right? And we have the glycerol. Okay. And that's that last step that we hadn't done before, by the way. So this is going to be, again, your triacylglycerols in your adipose tissue. These are going to be converted into your free fatty acids. Again, response to the hormone signals. This is your 7-TM receptor. Your hormone comes in. It's going to be, again, a G protein released. This is the adenylate cyclase pathway that we'll be going through. So the G protein is going to activate the adenylate cyclase. This is then going to convert ATP to CAMP. CAMP activates protein kinase A into its green active form here. And then what's going to happen is the protein kinase A is going to then take the HS lipase, and this is going to phosphorylate it to the HS lipase with that phosphorylate group, which is going to activate it. It's also going to act on the paralipin. It'll phosphorylate it again, and that'll give us the more active form of it. So we activate both those forms. Note those are going to then go on, and the paralipin is going to activate the ATGL right here by joining the CA group on it. This is actually what's going to activate then these other pathways. They're going to enzymatically activate them, move them along much faster. And so we're going to start converting the triacylglycerol to the diacylglycerol, and then the monoacylglycerol, and then the monoacylglycerol lipase is going to convert that final step down to the glycerol and fatty acid. And then we have three free fatty acids and a glycerol that we can then use. So your liver and lipid metabolism, the hepatocytes, these are your liver cells that function in the import, synthesis, storage, and secretions of your lipids. The processes that are going to be responsive to the diet and energy needs. The epinephrine CAMP pathway actually appears to regulate lipolysis from a lipid droplets to cultured lipocytes. This pathway, interestingly enough, is disrupted by ethanol. So you can actually disrupt this pathway by drinking. Wow, huh? Yeah, you don't often get that, do you? Yeah, I know. Albumin, this is the protein that transports fatty acids in your blood. It has 7 binding sites for the fatty acids of varying affinity. Your glycerol is going to be absorbed by the liver, phosphorylated, oxidized to the dihydroxyacetone phosphate, and isomerized to the glyceraldehyde 3-phosphate, which you may recognize from your other pathways, right? Remember, that's that sort of almost to the end, the doubling up, okay, of the glycolysis. So again, that's the glycerol right there. And you kind of probably predicted that if you remember your other pathways. Glycerol is, again, one of those kind of ubiquitous molecules, right? Note, by the way, you can, there's a very close derivative of this that can qualify for being a sugar, okay? So interestingly enough, guess what? Does anybody know how much sugars you actually have to eat in your diet? None. Yeah. Now, that's assuming your body functions properly, which is maybe 5% of humans. Frankly, the vast majority of us do not actually function properly. We have something that's not functioning properly in our biochemical pathways. So, the ATP and goes to ADP. This works on the glycerol kinase is going to be the enzyme. This is going to give us the L-glycerol 3-phosphate. And again, notice that phosphate has been joined down there. This is the L because it's chiral here. Okay. Right here, then, we have the glycerol phosphate dehydrogenase. That's the same enzyme we've actually done a few times now. This is going to take NAD+, going to NADH, and this is going to give us our dihydroxyacetone phosphate. You probably recognize the dihydroxyacetone phosphate, hopefully. Anyways, this is the triose phosphate isomerase. This is going to be the one that does that equilibrium between the two forms, which typically ends up converting over to the glyceraldehyde 3-phosphate, which is right here, which is the more typical form that gets used up more, right? Okay. So depending on metabolic needs, the liver processes glycerol by the glycolytic or the gluconeogenic pathways, depending on what you need. So here's your fat cell. It has the triacylglycerol. We're going to get our glycerols and typically 3 fatty acids, right? And then this is going to feed into the liver cell. Your glycerol is going to go into your liver cell. Your fatty acids can typically go into any cell that happens to need energy, okay? The fatty acids will provide energy as long as you have the oxygen requirements via the fatty acid oxidation going into the acetyl-CoA, and then into the citric acid cycle where it will fully oxidize. If it doesn't have the oxygen requirements, then you would go off on the branch pathways we had discussed for the same anaerobic processes. These right here, this is the liver cell. So if you just need energy, then you can just go down glycolysis further, right? We're at the final step of that doubling, that last step where it doubles in glycolysis, aren't we? And so you just go further down the glycolysis pathway, you'll get pyruvate. From pyruvate, you can again go into acetyl-CoA and do the citric acid cycle, or you can make tons of different things from that, right? Many options available. However, we have cells that are picky. They only get their energy from one thing, right? And so sometimes you need to go upwards and go into the gluconeogenesis to make glucose. And then you can take that glucose and you can distribute it around your body to feed things like your red blood cell, brain, etc., right? Yeah. And this is why you don't actually need any sugar in your diet. If you have fatty acids and glycerol, glycerol typically comes along with almost all the fatty acids, right? Or at least a very large number of them. You can actually do that. Though again, that does mean that you have to have all that pathway working perfectly, right? If you don't have all those pathway components, you're going to be in trouble. Okay? All right, so fatty acids are going to enter the intestinal cells through the FATPs. These are going to be transported within the cell by the FABPs. The acyl-CoA synthetase is going to catalyze the activation of the fatty acids through the formation of thioester linkages to the coenzyme A. This is going to require ATP. This is going to take place on the outer mitochondrial membrane. And so this is actually that pathway right here. Again, we have that fatty acid. Everybody comfortable with the fatty acid looking like that? That's that long chain right over here. That's the carboxylic acid part that gives it its acid. Okay. Anybody recognize that? That's the CoA, isn't it? Yeah, and it has a thiol group, which is where we link it for the acetyl-CoA. Okay. And so we're going to use up ATP. It's going to take off the pyrophosphate there and go to the AMP. And then this is going to link that up just like we would have any other acetyl-CoA. The acyl-adenylate, this is going to form when the fatty acid reacts with ATP. The formation of the acyl-adenylates when carboxylic groups are activated is actually a recurring motif in biochemistry. And we've actually seen this a number of times before, but we only now got to where we're making these things. Okay? And so, yes, biochemistry is like the Oberus, it eats its own tail. Just goes round and round, right? Okay. And that's the acyl-adenylate right there, by the way. Again, adenylate because of the adenine right here, sugar, the phosphate, and then that's the acyl group right there, right? Okay. The carboxylic group of the fatty acid is going to be bonded to the phosphoryl group of your AMP, and again, that's the AMP right there. The other two phosphoryl groups of the ATP is going to be released. Again, that's that pyrophosphate, that PPI. So step one, reaction of the fatty acid and ATP. This is going to yield your acyl-adenylate and the PPI. Step two, the reaction of the acyl-adenylate and CoA. This is going to yield your acetyl-CoA and AMP. And again, that's this right here. So we get the fatty acid reacting with the ATP. That's going to give you your acyl-adenylate right here. Again, this is an equilibrium. And this then is going to react with the CoA. This will give you your acetyl-CoA. Note, by the way, that this is not acetyl-CoA. Anybody know what the difference between those is? This is not going in the citric acid cycle. The difference here is number of carbons. Okay? So the acetyl-CoA just has those 2 carbons, right? The acetyl-CoA, this could be 30 carbons long. Okay? So this is not... Technically, it's not just one acetyl-CoA. Okay, we could actually get a bunch of acetyl-CoAs out of this if we spend the energy and time. Okay, huh? So this one right here, this is like a 30-20 carbon chain usually for these fatty acids. They're not going to be able to fit into the mitochondrial membrane because we don't have a transporter, typically. Yeah. Also, there's several other reactions that won't work as well when you had, instead of a primary carbon, secondary carbons. So you're going to have that problem too. Yeah. But technically, there are parts of that pathway that would work if you could get this to them. Yeah. And you could phosphorylate this and do certain parts of that pathway, which would be kind of an interesting thing. I wonder how often that actually happens, and you get these side products, which would be really unusual, side products. The partial reactions are going to be freely reversible, but the hydrolysis of the PPI is going to drive this overall reaction forward, and that's going to provide that energy drive for this. The biosynthetic reactions are going to be driven by the hydrolysis of PPI. Again, that's that recurring theme in biochemistry we've seen a few times, right? The overall reaction would be this process right here. Everybody okay with that? Fantastic. Okay, so carnitine, which is an alcohol with both positive and negative charge. If you remember from way back in the day when we talked about the amino acids, those are zwitterions, right? Does everybody know what zwitter means? It means two-handed. Okay? So, i.e., one of both type of ion. Okay? The fatty acids must be conjugated to carnitine to be transported across the inner mitochondrial membrane. The carnitine acetyltransferase 1, CAT1, is going to catalyze the transfer of the acyl group from the acyl-CoA to the carnitine. This is going to form the acetylcarnitine. It takes place in the outer mitochondrial membrane. And so right here is actually that transportation binding right here. Again, this is the acyl-CoA right here, carnitine, and we're going to bond it actually to the middle there, which is kind of interesting. But the acetylcarnitine is this right here. Can everybody see the zwitterion, by the way? All right, so the acetylcarnitine translocase is going to shuttle the acetylcarnitine across the inner mitochondrial membrane, exchanges the carnitine for the acetylcarnitine. The carnitine acetyltransferase 2, which is the CAT2, is going to catalyze the transfer of the acyl group from the carnitine to the coenzyme A. This is going to take place on the matrix side of your membrane, the reverse of the cytoplasmic reaction. Thermodynamically feasible because the O-acyl link in the carnitine has a high group transfer potential. Does everybody remember O versus N-acyl? N-acyl meant bonded to nitrogen, O-acyl meant bonded to the oxygen. Yeah. So it's just O-acyl. Okay. So again, this is that put into a nice diagram that looks easy and, it's beautiful. Okay, and so you have your acyl-CoA right here. Again, this is your inner membrane space. This is going to be your matrix side right here. And so the acyl-CoA is going to bind its acyl group, leaving the CoA free, to the carnitine to give you acylcarnitine. The enzyme that does that is carnitine transferase 1. This is then going to transport it across using the translocase. Great name, right? Translocase. Transporting these acylcarnitine groups across. This is going to deposit that on the matrix side. You will have the acylcarnitine worked on by the enzyme carnitine acetyltransferase 2. And this is going to rejoin that to a CoA, leaving the carnitine free, which can then get transferred across the other way in a displacement with the translocase to regenerate your carnitine at the beginning. The inability to synthesize carnitine actually contributes to the development of autism in some people, though again, remember that autism is a disease that can have many, many causes. Okay? This is just one pathway to that. We'll stop here on autism, apparently. Okay. You all have a great day. And I will see you all